Welcome back, students. Today we are going to discuss Unit 5, Multicellularity, Structure and Function Plant Life. And in this particular unit, we are going to discuss Mineral Nutrition Part 2. So before we start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are mainly going to discuss these points: nutrient deficiency diagnosis and significance, toxicity of micronutrients, mechanism of absorption of elements, ion movement and channels, soil as a reservoir of essential elements, nitrogen cycle and biological nitrogen fixation. Okay, so first topic, diagnosis and significance. Diagnosis of nutrient deficiency diagnosis. Okay, so deficiency of any type of element can cause multiple symptoms. In multiple symptoms, whether it is plant or animal, right? If there is a deficiency of any type of element, like in human, if iron deficiency is there, then that person feel tiredness, anemic. We can use the term anemic for the same. Similarly, if a type of element or more than one element is deficient in a plant, then that plant also show some type of symptoms. Okay, so that's why it is written over here ki deficiency of any element can cause multiple symptoms. Some symptoms can be caused by deficiency of different elements. Okay, and the diagnosis, that type of diagnosis require studying of all symptoms and comparing them with standard table. Okay, and different plant respond differently to the same element deficiency. This is another point. Different type of plant respond differently. Maybe one type of plant require magnesium in comparatively higher amount than other plant. Similarly for the iron. Okay, so that's why it is written over here, different plant respond differently to the same element deficiency. So to understanding these symptoms is a crucial point for agriculture and horticulture. Okay, so toxicity of micronutrients. As you know, the term micro is used over here. Micronutrient means the nutrient required in a very minimum amount, okay, or in a low amount. So if there is a moderate decrease of that micronutrient, that can cause deficiency symptoms. Moderately, if a particular type of nutrient is decreased in a particular environment, so that moderate decrease cause deficiency symptoms. And similarly, moderate increase, it will show toxicity to the plant. So if the concentration is low, that time plant body or a specific type of plant show deficiency symptoms. If concentration is high, micronutrient concentration high, that time plant show toxicity. Okay, so there is a narrow optimum concentration range. There is a narrow optimum concentration range. So now let's understand the term toxicity with reference to the plant. Okay, so this toxicity is defined as a mineral ion concentration that reduce tissue dry weight by 10%. Tissue dry weight by 10%. Okay, so toxicity is defined as a mineral ion concentration that reduce tissue dry weight of that particular plant by 10% is known as a toxicity. Okay, so critical toxicity concentration vary widely among micronutrients and plants. toxicity symptoms are difficult to identify. Excess of one element can inhibit the uptake of another one. Okay, you can understand this point with this example. Okay, as you can see in this particular picture, this is a healthy leaf, and these are magnesium. These leaves show some symptom with magnesium toxicity, or not magnesium, manganese toxicity. Okay, as this manganese toxicity show some kind of brown spots. These are the brown spots with chlorotic veins. Okay, why? Because this manganese, actually, if manganese is present in higher amount, higher concentration in the soil, that time it compete with the iron, also with the magnesium for the uptake and enzymatic binding. It want, if concentration is high, then it want to bind with the same enzyme, with all those type of enzyme which actually normally bind with the magnesium and iron. So it show a competitive behavior. Okay, so that's why it is written over here that manganese compete with iron and magnesium for uptake, right, and enzymatic binding. And it also inhibit calcium translocation into the shoot apex. So if this manganese is present in high amount in a given soil, that time it actually reduce the concentration of iron, magnesium and calcium, right? So excess magnesium, if concentration of manganese is high in a particular environment, it may induce deficiency of iron, magnesium and calcium. Okay, so symptom of manganese toxicity may actually be deficiency symptoms of these three: iron, magnesium and calcium, because it show a competitive behavior with these metals. So we can also say that this magnesium toxicity actually be deficiency symptoms of other elements like iron and magnesium. Clear? Okay. Now, mechanism of absorption of element. So, research on absorption mechanism has been conducted using different isolated cells, different tissue or organ. So, there are two phases of absorption. Two phases. One phase is known as apoplast, another phase is known as symplast. Okay, so first phase is known as apoplast. This is the initial phase, and this one is the rapid uptake of iron into the free space, or we can say outer space of the cell. Initially, very rapidly, cell actually take all the soils, all the sap which is present just outside the plant body in the soil. So that's why initial uptake is very rapid. Okay, initial uptake of ions is very rapid into the free space or outer space of the cell, and this is known as apoplast, and this phase is passive. Cell doesn't need to spend a kind of energy for this, as concentration of water is high outside environment, and so very rapidly cell just show a rapid response, rapid uptake of the iron in the free space. Okay, after that there is a slow uptake of those ions into the inner space of the cell. Okay, and this is slow uptake is known as symplast, and this particular phase require energy, require metabolic energy. That's why this phase is active. So symplast require energy, means it will use ATP. Okay, and this passive phase doesn't require any kind of energy. Okay, So apoplast phase in which apoplast is involved, this phase is passive. A phase in which symplast is involved, this one is active phase. Now, ion movement and channels. This passive movement into the apoplast occur through some ion channels, which are transmembrane protein. Those are also having selective pores also. Okay, so passive movement into the apoplast occur through ion channels, which are present in the as a transmembrane protein, and those are also acting as a selective pores for selective ions. Entry or exit of the ion to and from the symplast is an active process, require metabolic energy, which already I told you earlier. Okay, so entry and exit of ions to and from the symplast is an active process which require metabolic energy. And this ion movement inside the cell and outside the cell, this process is known as flux. If ion come inside the cell, or we can say inward movement of the ions, that time we can use the term influx for the same. Inward movement inside. Okay, and outward movement is there, that time we can use the term efflux for the same. Okay, efflux for the same. Now, translocation of solute. Translocation of solute. As you know that mineral salt are translocated through xylem along with the ascending stream of water. Okay, as these root hairs, through root hairs, root absorb water from the soil and through xylem vessels. Okay, and take it out other parts of the body where it is required. Okay, that's why there is a transpiration pull occur. The water is pulled up through the plant via transpiration pull, because transpiration is a continuous process. Water is lost by the leaves through different metabolic activity. So continuously root also absorb water from the soil. So there is a transpiration pull developed. So with this transpiration pull, water is pulled up, right, to the other parts of the body via roots. So analysis of soil sap also show the presence of mineral salt in it. With water, with water, root also absorb some minerals from the soil, right? So that's why it is written over here. Analysis of xylem sap show the presence of mineral salt. Use of radioisotopes of minerals elements support that they are transported through the xylem. Soil as reservoir of essential elements. So, as you know that weathering of rock, right? Weathering of rock, breakdown of rock actually provide different kind of inorganic salts to the soil, and weathering is a process by which that particular rock or rocky area develop soil type, different type of soil type. Okay, or this weathering process or breakdown of soil enrich the soil with dissolved iron or inorganic salt, which we further use the term mineral nutrients, right? So essential nutrients or essential minerals, as already in the part 1 we have discussed that some minerals are comes under essential category. Some are required in a large amount, known as macronutrients. Some are required in low amount or micro amount. Those are known as micronutrients. So, but both the type of nutrients, macro and micro, both are require required by the plant for their healthy growth. So, essential minerals are crucial for crop yield. Both macronutrients Macronutrients means nitrogen, phosphorus, potassium, sulfur. Micronutrients like copper, zinc, iron, manganese. Okay, these are micronutrients. These are the component of fertilizer and are applied based on the plant seed. But both of them means macro and micro are important for the healthy growth of a plant. So, and these all the type of nutrients are present in the soil, right? In the soil, and from where it comes from the weathering of the rocks. So, what is the function of soil? The soil is the main support system for the plant, which actually provide different kind of minerals to the plant. So it supply minerals to the plants. It also harbor nitrogen fixing bacteria and other microbes also. Okay, and other microbes also. Soil also hold water. Okay, soil supply air to the root, right? And soil act as a stabilizing matrix for plants. So these are the functions of soil. Why soil is important for the plant? Not only um, not only for the purpose that soil is having water holding capacity, soil also contain different kind of minerals, which is utmost important for the plant growth. It also having different kind of bacteria, plant growth promoting bacteria, right? And it also having nitrogen fixing bacteria, right? So these are other important functions of the soil. Now let's discuss nitrogen cycle. So first of all, we have to understand the importance of nitrogen. Why we need to discuss this nitrogen cycle? Because this nitrogen is essential. Why nitrogen is essential? Because nitrogen is present in various amino acids, means various kind of proteins. Proteins are made up of amino acids, and most of the hormones are proteinaceous in nature, right? Most of the vitamin also contain nitrogen. Chlorophyll is also having nitrogen, right? So that's why nitrogen is very important element for the plant and also for the animals, right? But plant and microbe compete for limiting soil nitrogen. Both of them are plant roots are present in the soil, and so many microbes are present in the soil. So both of them compete with each other for limiting soil nitrogen, making it a limiting nutrient in ecosystem. Okay, as you can see, this is the structure. Ammonia, this is the nitrogen, this is the carboxylic group, and it is the basic structure of any amino acid. So there is a nitrogen fixation process also, nitrogen fixation, in which this nitrogen, in nitrogen, two nitrogen atoms are attached to each other or joined to each other with triple covalent bond, right? So nitrogen exists as a two atom joined by strong triple covalent bond, and the conversion of this N2 into this NH3 or ammonia is in the presence of lightning, or UV radiation, it will convert into various type of nitrogen oxide also, like NO, nitrogen dioxide, dinitrogen oxide, and so, right? So conversion of nitrogen to ammonia is termed as nitrogen fixation. Nitrogen fixation means conversion of nitrogen to ammonia. Nitrogen fixation, right? Okay, so what are the natural sources? Lightning, as you can see in this particular picture, lightning. UV radiation. In the presence of lightning and UV radiation, normally this nitrogen get converted into various nitrogen oxides like NO, NO2, N2O. Okay, so this is comes under natural sources category. Another category is human sources, in which industrial combustion occur, forest fire occur, automobile exhaust, power station. These are the sources in which nitrogen convert into various oxides form, various nitrogen oxide form. Now, another subtopic related to this nitrogen cycle is ammonification. Ammonification means decomposition of organic nitrogen which is present into the dead plant, dead animal into ammonia. Means organic nitrogen to ammonia. This is known as ammonification. When atmospheric nitrogen, this N2 gets converted into ammonia, that is something different, nitrogen fixation. Right? But when this organic nitrogen, conversion of this organic nitrogen into ammonia, that time we can use the term ammonification, in which decomposition process occurs, decomposition of organic nitrogen occurs. Right? As you can see in this particular picture, this is the plant, this is another plant. There are certain nitrogen fixing bacteria present into the soil, right, which fix the nitrogen, and this nitrogen is used up by the legume plants and other plants also, right. And this ammonification means nitrogen fixing soil bacteria are there. When these nitrogen fixing bacteria fix the organic nitrogen into ammonia, convert it into ammonia, and further this ammonium ion is decomposed by the a different kind of bacteria or fungus strain. Organic nitrogen is present when this plant will get die after some time. That time the organic component, organic nitrogen which is present in this plant body is decomposed by various bacteria and fungi and convert it into the ammonium ion, and further some nitrifying bacteria convert this ammonium ion into nitrite and nitrate, right. So this process is known as ammonification. Further conversion of this ammonium ion into nitrite is known as nitrification, nitrite and nitrate, right. This is known as nitrification, and by further this nitrate is further or nitrite further is used up by the plants if these are present into the soil. Okay, and if these nitrite get converted into the atmospheric nitrogen by denitrifying bacteria also. So this is a nitrogen cycle, how nitrogen move from atmosphere to the soil in plant body, and then further various used up by various aerobic, anaerobic bacteria or fungal strain, and then conversion of this ammonium into nitrite and nitrate, and further with the help of denitrifying bacteria it will just used up by the denitrifying bacteria, and then again released as a atmospheric nitrogen into the atmosphere, right. So normal nitrogen, atmospheric nitrogen, this nitrogen get fixed into the various kind of nitrite or nitrate in the presence of different kind of bacteria. Then this is known as nitrogen fixation, right. And decomposition of organic nitrogen, this decomposition of organic nitrogen, this process, this is known as ammonification, right, in which some ammonia re-enter into the atmosphere. This ammonia re-enter into the atmosphere by converting into nitrite, this nitrite or nitrate by some nitrifying bacteria. Clear? Okay. And what is this nitrification? Same conversion of ammonia into nitrite or nitrate, right? This is known as nitrification. So what is written over here? Ammonia oxidized to nitrite by some bacteria known as Nitrosomonas or Nitrococcus. Further, this nitrite oxidized into nitrate by nitrobacteria, means NO3, ammonia to NO3, right? Nitrococcus or Nitrosomonas, and then further NO3 to NO2, right? By nitrifying bacteria. Like chemoautotrophs. These two processes known as nitrification. Clear? Ammonia to nitrite and then nitrite to nitrate. Ammonia to nitrate and then nitrite to nitrate by various bacteria like Nitrosomonas, nitrobacteria. Right? Okay. This is known as nitrification. So there are three terms: nitrogen fixation, ammonification and nitrification. All these 3 points are totally different. After that, absorption and assimilation. Absorption and assimilation. So plant actually absorb nitrate, NO3, okay, and transport it to the leaves, reduce it into the ammonia, which form amino group of amino acids. So plant absorb nitrogen as nitrate, NO3, and then transport this NO3 to the leaves, which reduce it in ammonia and further which form amino group of amino acids. Now, what is denitrification? Nitrate, NO3, which is present into the soil, is reduced to nitrogen gas by Pseudomonas and Thiobacillus, some bacteria which is present in the soil, returning to the nitrogen into the atmosphere. Means this NO3 to N2. This is known as denitrification in the presence of Thiobacillus or Pseudomonas. This process is known as denitrification. Clear? So this process, denitrification, nitrate, right, to atmospheric nitrogen in the presence of denitrifying bacteria, means Pseudomonas. This process is known as denitrification, right? So this process is known as atmospheric nitrogen to this nitrogen fixation. This process is known as nitrogen fixation. Nitrogen fixation also occur in the presence of bacteria, right? And or via normal in the lightning process or in the this nitrification, nitrogen fixation, right? And this decomposition means or conversion of organic nitrogen into ammonia, right? Conversion of organic nitrogen into ammonia, this is known as ammonification, right? And then conversion of this ammonia into nitrite, then further nitrate, this is known as nitrification. Clear? Now, next topic is biological nitrogen fixation. Biological nitrogen fixation, in which reduction of nitrogen to ammonia. Reduction of nitrogen to ammonia by living organisms is known as nitrogen fixation. in which an enzyme known as nitrogenase enzyme is involved, which actually catalyze this process in the presence of some nitrogen fixer bacteria. There are various kind of free living nitrogen fixed bacteria. Some are aerobic in nature, some are anaerobic in nature, like Azotobacter and Bizarnikia. These are aerobic in nature. Anaerobic Rhodospirillum bacillus. These are anaerobic in nature. Some cyanobacteria like Anabina, non-stalk are also involved in nitrogen fixation. These all are comes under free living nitrogen fixing bacteria. Okay, so reduction of atmospheric nitrogen to ammonia in the presence of some bacteria, okay, and some enzyme like nitrogenase. This is known as biological nitrogen fixation, as you can see over here. Okay, nitrogen, this is the atmospheric nitrogen. It will fix into the organic nitrogen in the presence of nitrogen enzyme and some bacteria. And this is known as symbiotic nitrogen fixation. So in previous slide, free living bacteria involved in this nitrogen fixing process. Free living bacteria. Some are aerobic, some are anaerobic, or some cyanobacteria. Here, some bacteria show a symbiotic relationship with some plants, mainly pulses, legumes. Okay, that comes under symbiotic nitrogen fixation category. Okay, so in which this Rhizobium species is the main type of bacteria. This actually associate with the roots of legumes like alfalfa, sweet clover, sweet pea, lentils, garden pea, right? Mainly pulses, as I told you. So these there are symbiotic relationship between the roots and bacteria. So, and this is the common association present in the root nodules, and these are shown as a small outgrowth of the root. This is known as root nodules, in which symbiotic bacteria are present. These are the root nodules. Okay. Non-leguminous plants are also there who are able to fix atmospheric nitrogen, in which a microbe known as Frankia involved in fixing nitrogen in non-leguminous plant. So Frankia is a type of microbe which form nitrogen fixing nodules. This is the nitrogen fixing nodules. Okay, and these are present in the non-leguminous plants. And this is the example, Elnus. So both Rhizobium and Frankia can fix atmospheric nitrogen as symbiont, though they are free living in the soil. But Rhizobium is present in the legumes or pulses, roots of the pulses or legume plants, and Frankia is present in the non-leguminous plant as a symbiotic relationship. So this root nodule appear as a spherical or spherical outgrowth on the soil, which are noticeably just before the flowering. Cutting through a nodule, if you will cut these nodule, it will reveal a red or pink center portion. Why? Due to the presence of leguminous hemoglobin, which is known as leg hemoglobin. This leg hemoglobin is present into the nodules. Now, let's discuss how these nodules form. Okay, so this Rhizobium bacteria continuously multiply and colonize near the root. Okay, and attaching to the epidermis and root hairs of the cell. As you can see over here, this is the free living Rhizobium. These are the rhizobia. Okay, continuously multiply and form colonies and attach to the epidermis of the cell, plant root, and at the same time this plant roots release certain kind of flavonoids as chemical signals to attach rhizobia. Okay, and rhizobia also secrete a kind of factor known as NOD factor. So this infection thread, this is a thread, infection thread is formed when plant root release a kind of chemical to attract the bacteria. Continuously this infection root is formed, and other PGPB means plant growth promoting bacteria may induce nodulation process, and after that this infected thread grown into the cortex. It also grown through the cortex side. This is the epidermis, dark one, and then this infected thread grown into the cortex of the root and release rhizobia. And this is how root cells divide to form nodule, while rhizobia become bacterioid. PGPB also colonize into the root nodules. So these are the steps. Okay, it is also written over here. Initial, what is the first step? Rhizobia bacteria multiply and colonize the root surroundings, right? Attached to the epidermis and root hair cells. Epidermis and root hair cells. Then an infected thread carries bacteria into the root cortex. This infected thread carries the bacteria into the cortex. This is the cortex part, right? An infected thread carries bacteria into the root cortex, initiating nodule formation, and then bacteria are released into the cell. These bacteria are released into the cell, differentiated into the specialized nitrogen fixing cell. This is how nodule form, right? The nodule establish a vascular connection with the host plant for nutrient exchange. Nodule contain biochemical components like nitrogenase enzyme and lag hemoglobin. Okay, these nodule contains nitrogenase enzyme and lag hemoglobin. And this nitrogenase and oxygen, this nitrogenase enzyme, which is a molybdenum iron containing enzyme, this particular enzyme actually catalyze the conversion of atmospheric nitrogen into the ammonia. Atmospheric nitrogen into the ammonia. Okay, and this particular enzyme is sensitive. This particular enzyme is sensitive to oxygen and require anaerobic condition. This enzyme require anaerobic condition. This one is sensitive to oxygen. Nodule also contain lag hemoglobin, which is a oxygen scavenger. So this lag hemoglobin very quickly utilize the oxygen, and this is how it protect the enzyme. Okay. Now microbial adaptation, free living rhizobia. These are aerobic in nature but become anaerobic during nitrogen fixation to protect nitrogenase enzyme. And nitrogen fixation require a high energy input, means 8 ATP molecule per ammonia molecule. Okay, sourced from the host cell respiration. Now ammonium ion formation. At a particular physiological pH, ammonia is protonated to form ammonium ion, and these ammonium ion are toxic to the plant and can't accumulate it. Okay, so this ammonium ion very quickly react with alpha ketoglutric acid and form glutamic acid. This ammonium ion is very toxic, so this is very quickly utilized by the alpha ketoglutric acid in the presence of glutamate dehydrogenase, and NADPH is utilized as an energy source, and this alpha ketoglutric acid then convert into glutamate and water is released. So this process is known as assimilation into the amino acid. Now transamination. So the transfer of this amino group Transfer of is this amino group from one amino acid to the keto group. This is the keto group, is known as transamination, in which glutamic acid is the primary amino acid. This is glutamic acid, and it is involved for the transfer of NH2, forming other amino acids. Okay, and this process, this reaction is catalyzed by transaminase enzyme. Transaminase enzyme. Clear? Now, formation of amides, asparagine, glutamates. These asparagine form aspartic acid and glutamate form glutamic acid respectively by adding amino group. Hydroxyl part of the acid is replaced by NH2 radical, and these amides contain more nitrogen than amino acid and are transported by xylem vessels. Now, export of fixed nitrogen nodules of some plants like soybean. Nodules of some plants like soybean export fixed nitrogen as ureates. Ureates have a high nitrogen to carbon ratio and are transported with the transpiration streams. These ureates have high nitrogen content, high nitrogen to carbon ratio, and are transported with the transpiration streams. So this is all about the today's video lecture. I hope most of the things has been cleared now. Still you have any query, any doubt related to today's lecture, please let me know. We will discuss it later on. Till then, take care, bye bye, and keep learning.